here is gray matter, the lighter staining areas are white matter. The gray matter is separated into regions that we call horns. At the bottom sort of right corner of the picture, we have a region called the ventral horn. At the top of the picture, that gray matter is what we would call the dorsal horn. Ventral means anterior, and dorsal means posterior. So the ventral part would be the side of the spinal cord closest to the belly. The dorsal part would be the side of the spinal cord closest to the back. This particular section doesn't actually have a distinct lateral horn, but see that little that little white arrow off to the side, which is actually how I would move in this website from picture to picture. The gray matter closest to where that white arrow is, in a different part of the spinal cord, would be the lateral horn. In this particular area, there actually isn't a distinct lateral horn. The three areas are related to different functions. The ventral horn, which is what we're going to look at, contains neurons that control skeletal muscles. So the ventral horn is full of motor neurons, which we learned about last section in the musculature. The motor unit is comprised of one neuron and all of the muscle fibers it controls. The motor neurons that make up motor units are these neurons that we're going to be looking at here. The dorsal horn, which we are not going to be looking at today, contains neurons that are specialized for sensory reception. Now, they're not the ones that are actually getting sensory stimuli directly. They're receiving information from peripheral nerves, carrying that information in. And we're going to look, after this slide, we're going to look at another slide that is those types of neurons that do actually receive the sensory stimuli directly. So we'll get to that next. The lateral horn, which isn't here in this picture, it's okay because that's autonomic control in your spinal cord. Wait, again, autonomic system is something we're going to talk about in A&P too. But so we're going to look at some of these cells down here in the ventral horn a little bit more closely. Before I go to that, let me just remind you of, oops, where is it? This picture here, the three different types of neurons. What we're going to be looking at are multipolar neurons. Okay. The other slide that I just mentioned that we're going to get to later on that has to do with sensory stimuli, those will be unipolar neurons. Okay. We will not be looking at bipolar neurons this semester because they only have to do with subjects of A&P 2. So, but this is what we're going to look at here. So this is the 4x objective spinal cord slide. Then this is the 10x objective spinal cord slide, specifically of the gray matter. I have a separate one for the white matter, which we're not going to look at right now. So now we're in the ventral horn area. So before we could see some gray matter and some white matter, now we've zoomed in to part of the gray matter, and specifically in that ventral horn. We can see these nice big cells that stain very distinctly. These are the neurons that we're interested in. Okay? Now, the staining procedure that's used here is a special stain that's special for neurons. And I'm going to introduce its name, but its name isn't that important. I just want you to understand that what it's doing is on showing us certain things on purpose. It's what's called a Nissl stain, N-I-S-S-L, weird little name. It's the name of a scientist who, I don't remember exactly, but I think he's from somewhere in the Eastern Europe area originally. He's of course dead now. But so it's some sort of Hungarian or Slavic name. I don't remember exactly where it comes from. But it's a variation on staining. Most of the stains we look at, we usually see a nice dark nucleus, and then the cytoplasm will be a lighter color, and then extracellular space will usually be very likely if stained at all. In a Nissl stain, actually what we see is in neurons, the cytoplasm stains darkest, and the nucleus usually is a little void in the middle. Okay. So let me go to the 40x subjective version of this, which will actually zoom in on these cells right here. That's not it. Okay, I got to go back to the list to find it. Okay, there it is. So you might recognize this cell that was just in the previous one, and then this one next to it. This is a great example of what a Nissl stain shows us in a neuron. The nucleus is pretty much a clear void right in the middle, and then the cytoplasm stains pretty well, but it's usually a kind of mottled or textured pattern. So it's not a solid blue color. There's some gradations, dark and light patches within it. Okay. Now, you can see in the middle of the nucleus a very distinct dot, which is what's called the nucleolus. Now, the word nucleolus translates to little nucleus. So somebody looked at that and said, hey, there's obviously the nucleus, but then it has a little dark spot in the middle of it, so let's call that the little nucleus. That's where the word came from. What the nucleolus is it's the part of the nucleus where there's DNA that codes for ribosomes. Okay? So it contains the genes for ribosomal RNA. Okay. Ribosomes are made out of RNA. So the gene is transcribed, so the DNA is copied into RNA, like any transcription is going to do. But for ribosomes, that's the end product. That RNA that's transcribed from the gene is what becomes the ribosome. And so the ribosomes are assembled in the nucleolus at that particular point. And that's why it's such a dark little spot. There's a high concentration of RNA. The DNA, because of how Nissl staining works, doesn't stain very darkly because there are things offsetting its particular negative charge. But the RNA that makes up the ribosomes picks up a lot of stain because it's actually very negatively stained. And the point of this procedure called the Nissl stain is to highlight ribosomal RNA. Now, what do ribosomes do? Anybody? What do ribosomes do? They make protein, right? So they translate RNA into protein. The messenger RNA from other genes gets translated into protein because the ribosome is sort of this super enzyme that reads the genetic material and then takes amino acids and joins them together with peptide bonds. Okay. Where do ribosomes do that? Where do we find ribosomes in the cell? In the cytoplasm. Most of them are associated with what?
Endoplasmic reticulum.
Right. And which version of endoplasmic reticulum?
Rough.
Rough. And it is rough because it's got a bunch of ribosomes stuck all out on the outside. Smooth endoplasmic reticulum doesn't have ribosomes. So in the cytoplasm, there are some free ribosomes, but then most ribosomes are associated with the rough ER. The Nissl stain, the reason we can see that cytoplasm so clearly is because it's staining those ribosomes. And the sort of texture, pattern, whatever we want to call it, of this cytoplasm is really showing you where there's higher concentrations of ribosomes and the lighter spots are where there aren't a lot of ribosomes. Okay. So to some extent, what we see in that Nissl stain is sort of a picture of where the rough ER is. You may hear reference to Nissl bodies. That's just a microscopist's name for the rough ER, because rough ER stains and a Nissl stain. So when you see them, or if you see Nissl bodies, it's just saying that texture pattern that we see inside the cell. Now, if ribosomes make proteins, what does it tell you when we see a cell that stains so darkly for specifically ribosomes? What is important about that cell? Ribosomes make protein. Dark stain saying lots of ribosomes. What does the cell do? Sure, you're all being silent because you're like, oh, it couldn't be that simple and obvious of an answer. But it is. Ribosomes make proteins. These cells stain darkly for ribosomes. So these cells do what? Make protein. Right. Now, the cells that we're going to concentrate on looking at nervous tissue are usually fairly large neurons. Okay? And these particular ones are the neurons that control muscle fibers. So this might be a neuron that controls the muscle that flexes my fingers. Okay? And so the neuron is located probably around the base of my neck. Okay? The axon extends out from the spinal cord, goes in a nerve in my arm all the way out to where the muscle is to make it contract and flex my fingers. So that might be 45 to 60 centimeters long. The cell body itself is 100 or so millimeters in diameter. Micrometers, 100 or so micrometers in diameter. Nope, that doesn't sound right. It's really small. Let's just leave it at that. I'm getting the words mixed up in my head. But very small diameter cell, very long axon. And that axon needs proteins all along its length. Okay, there's cytoskeletal proteins that give the axon its shape and strength. And there are ion channels and enzymes and neurotransmitter receptors and all sorts of things that are in that cell that have to be made. All of the proteins that serve the entire cell all the way out to the end of its axon are made in the cytoplasm here. So it's a really big cell with lots of ribosomes because it's making a lot of proteins for all of this cell to do what it needs to do. Now we don't really see dendrites sticking off of this so clearly, and we can't really tell where its axon comes off. The reason for that is because the cytoplasm in the cell body is what we're staining. The, quote, cytoplasm in the dendrites of the axon, and I say, quote, cytoplasm because it doesn't contain everything that cytoplasm contains, doesn't stain. In fact, we don't call it cytoplasm. It's in the dendrites, we call it dendroplasm, and if it's in the axons, we call it axoplasm. Neither of which stain with initial stain. So we don't see that part of the cell. We just see the cell body itself and all of its cytoplasm, which has a lot of ribosomes in it, which are responsible for making all the proteins that are going to go into the dendrites and down the axon and all over the cell. It has to be a really big cell to support that. Now, I use the example of this might be a cell that controls flexing my fingers because the slice that we're looking at, the cross-section of the spinal cord we're looking at, is from the cervical region, the neck region, which we control with the arms. But imagine if we were looking at a cross-section of spinal cord towards the bottom of the spinal cord, which is responsible for controlling muscles in my foot. So we'd have a really big cell there that has an axon that goes all the way down from about the second lumbar region of my spinal column through my leg to my ankle, and it turns and goes forward in my foot to control whatever muscles in my big toe, for instance. That axon is probably going to be like a meter in length, if not more, for somebody of my height. So again, really big neurons because they have really long axons. Okay. Now, this cell next to the one I've been talking about, so I've been talking about this cell right here. This one off to the side, we don't really see clearly where its nucleus is because we've not probably cut through the middle of the neuron, but we can see a fairly distinct branch going off this way, and the hint of something probably coming off this way. and there might be something coming off of this bump right here. So we can see stuff, where stuff must be coming off of this. And this particular neuron, for whatever reason, the base of its dendrites actually contains some cytoplasm, so it picks up some stain. But we can look at that and say, okay, there's definitely 3 things coming off of that. And remember, anything more than 2 makes it a multipolar neuron. The other cells here, you can't really see stuff coming off of them. And largely that's because the dendroplasm and axoplasm doesn't stain. But that doesn't mean that they don't have those things sticking off. In fact, they do. You will not be required in this course to identify a cell and say, is that unipolar, bipolar, or multipolar? Because the answer is going to always be very obvious. Except for the next slide I'm going to show you. Any neuron that we look at this semester is a multipolar neuron. Might not be able to see all the dendrites and axon on it, but it has to be multipolar because that's what we're looking at this semester. We will not be looking at bipolar cells this semester, and the unipolar cells we look at this semester will be only in the next slide I'm going to show. So these big cells we see here are all versions of multipolar motor neurons in the ventral horn. Now there are some smaller cells in here. So we can see a nucleus there and maybe a little hint of cytoplasm around it. Nucleus here, a little hint of cytoplasm. Another nucleus, maybe that's some cytoplasm. But then we also see some dark spots here, which are probably nuclei of cells, but we don't really see any cytoplasm around them. The ones that have nuclei that you can kind of really make out a nucleolus in are probably very small multipolar neurons. And they have dendrites that maybe extend only a few micrometers out from the cell body. And the axon can probably also be measured in micrometers. So those are very, very small neurons compared to the big ones that have axons that can be measured in centimeters or even a meter. Okay. The ones that are really dark nuclei that don't have a very clear nucleolus, I'm not positive about this because this is, again, not my expertise, but this dark spot here is a nucleus. That might be the nucleus of a glial cell. And if we're looking at gray matter like this, most likely it's an astrocyte. There's no way to be sure because this type of stain doesn't give us any details. I can't even tell you for certain that that's a glial cell. But assuming it is a glial cell because the nucleus looks different from the neurons that we've been talking about, in this part of the nervous system, gray matter of the central nervous system, the best bet is that it's an astrocyte. It could be an oligodendrocyte, but those are more likely going to be in the white matter. It could be a microglia, but I doubt it because of 2 reasons. One, microglia are very small relative to the other cells, so that's a pretty good sized nucleus, so I would expect that's not a microglia. And 2, microglia are, for the most part, fairly rare. In this field of view, I'd be surprised to even find 1 microglia in here. So it's not likely to be a microglia, but we really don't have any real data to say what type of cell that is. It's my best guess is that it's an astrocyte. Now, the next slide I want to show you is of what's called the dorsal root ganglion. So earlier when I was defining ganglia, I showed you on this vertebral column model with the yellow rubber nervous system inside of it, that the base of the nerves coming out of this is a little thickness, which is a ganglion, which is what we have on the slide right here. Now, on the poster that I've been pointing at earlier, I showed you the cross-section of the spinal cord here. In this same frame, this little egg-shaped thing off to the side is that ganglion. Now, I've labeled this, and I call this a dorsal root ganglion. On the poster, the frame right next to the spinal cord cross-section says posterior root ganglion. Those are 2 names for the same thing. Dorsal and posterior are interchangeable names for directions as far as we're concerned here. Dorsal means towards the back, and the posterior part of the spinal cord is going to be the backside. So posterior and dorsal mean the same thing. Now, nerve roots are referring to where the spinal nerve, the thing that comes out and goes in the periphery, where it attaches to the spinal cord. There's actually two roots, one on the ventral side, which are axons leaving the ventral motor neurons going out to the periphery, and another root on the dorsal side, which are axons of these cells entering into the spinal cord. And so attached to the dorsal root is a ganglion where the cell bodies that make up the dorsal root fibers are located. In this field of view, which is at 4x magnification, there happens to be a fairly obvious cell body right there. For whatever reason, it stains a little dark and it stands out compared to its neighbors. If you look closely, there are other cell bodies. There's one here, it's kind of light and we don't see its nucleus, but it's about the same size. So there are some there, and then there's some in this dark patch down here. For whatever reason, this one just happens to stand out at 4x pretty clearly. If we go to the 10x version of this picture, I believe this is that dark one that I just pointed out. And part of the reason why it probably stands out as such a dark staining cell, besides the fact that its cytoplasm for some reason is a little darker, is also because it's surrounded by some white space. I don't know, the tissue might have torn right around the cell or something like that. And so that contrast really leaps out to our visual system. But this cell right here and the one right next to it are about the same size. And we can see that there's a little nucleus, and we can see the dot of its nucleolus in the middle there. Because of how this one that I've been pointing out stained kind of weird, it doesn't look quite the way that we expect. But those are both cell bodies of really big neurons. Now, these neurons, the ones that are in this ganglion or in ganglia like it, are unipolar neurons. We will not see on any of these neurons the one axon coming off of it. It's just the likelihood of slicing through the cell in just the right place to see that one spot that something comes off is unlikely. So we'll just see big round cell bodies. But understand, somewhere there's an axon coming off of this, which comes out into this area and splits, and half of it goes one way and half of it goes the other way. Now, again, it's sort of my own personal convention. I usually think of the left side of the picture as the side that's going to the periphery, so where the dendrites are going to be. And then the right side of the picture is going into the central nervous system. I don't actually know how this particular ganglion is oriented. It doesn't really matter, but that my way of describing it kind of keeps the story up. Unipolar neurons, which we find in ganglia like this, are pretty much always going to be sensory neurons, which means their dendrites are directly getting sensory stimuli. Most of them are going to have dendrites in the skin, in the dermis in the skin. So the free nerve endings or the encapsulated nerve endings in the skin are going to be dendrites of these things. So free nerve endings will receive information like pain and temperature, and then other dendrites that have some supporting cells around them will be responsive to pressure and vibration and touch, which is kind of pressure. The Merkel cells that are in that basal layer of the epidermis connect to some dendrites of these cells, making the Merkel disc, which is responsive to light touch on the surface of your skin. So these are all cells that are receiving those stimuli from the skin or from other structures in related senses. So stretch receptors in your muscles, in your tendons, or other receptors in your joints that can feel the direction that your joint's moving. Those cells are all these big cells that we see here. The bigger the cell, the longer the axon. So I suggested a little while ago, imagine a neuron at the bottom of my spinal cord, and its axon goes out to my big toe to cause a muscle contraction in my toe. Imagine now a neuron whose dendrites are in the tip of my big toe. So when I run into something with my toe and I stub my toe, that sensory experience, the pressure of my toe against the foot of this table here, whatever, that's going to go from the dendrites in my big toe all the way back in my foot to my ankle, then turn up, continue up my leg to my spinal cord. And actually, it doesn't end there. It continues. The axon that started in my big toe still keeps going up my spinal cord and finally synapses in my brain, at the base of my brain, about here in my neck. So if that motor neuron controlling the muscle in my big toe is about 1 meter long, the axon carrying the information when I stub my toe up to my brain is probably about 2 meters long. Really, really long axon, really, really big cell body. Okay, so we're going to see those. Now, if we look at this a little bit closer, you will see a little bit of variation in cell body size. Not all of them are huge, because not all of them are going to be carrying information that far. But there is some variation. So the two slides that I just showed you were the 4X and 10X dorsal root ganglion cells. For the 40X version of this, I titled it unipolar cell because we're looking at one particular cell. Now, I'm almost positive this is not that dark cell that I pointed out before, but I didn't really pay too much attention when I was taking these pictures exactly where on the slide it comes from. But let's say that's about the size of the cell that we're looking at. There are smaller looking cells alongside of it. The fact that we don't see a nucleus in these cells probably means that we're not cutting through the center of those cells. Okay, so we might be sort of slicing off the edge of the cell or something like that. But there is some variation in cell size. And actually, if we go back to the 10X version, if you look closely, say like down here, we can see the nucleus in that cell. So we must be cutting pretty much through the middle of it. And that cell is definitely smaller than this cell up here. So again, size probably is directly related to the length of the axon, what this particular thing is connected to. Not all of these cells are going to carry information all the way up to the brain directly. Some of them carry information just into the spinal cord right there so that it doesn't continue on any further. And so some of these smaller ones might be like that, and they're usually going to be pain receptor neurons or something like that. So I was describing that really long axon. I said stubbing my toe, but I didn't mean the pain of stubbing my toe. I meant really the physical contact, the pressure, because that kind of information would be carried up a really long axon directly to the brain. Pain information would be carried by a shorter axon and go just right into the spinal cord, and the axon would stop there. But we see some variation in size. Now, let me go back to that 40X objective, because I want to point out some glia here. Now, there are two types of glia in the periphery. We're in a ganglion. A ganglion is a peripheral structure. The two types are called satellite cells and Schwann cells. Satellite cells are named satellite cells because they look like they're orbiting the big cell bodies of these neurons. Okay? So these could be little moons orbiting around this big planet in the middle. Okay? Satellite cell. They act as supporting cells. Now, there's a picture in the book that shows a more three-dimensional representation of these cells together, and the satellite cells kind of make an outer coating around the whole cell. So they kind of protect the cell from what's around it. They do not make a blood-brain barrier because we're not in the brain here or in the central nervous system. But they do have similar kind of protective, supportive functions as astrocytes that I talked about earlier. Then up here we have a region that's not neurons, but rather the axons. If I back out to the 10X, so this big piece right here, you can really tell that there's just fibers running through this area, as opposed to the cell bodies here, the cell bodies here, or the cell bodies here, here, here. So these big chunks right here, this is actually some of the nerve that's passing through the ganglion. Okay, these are all axons. So the cell bodies that we see in here with those fibers, with those axons, are probably the nuclei of Schwann cells, which are the ones that insulate axons in the periphery. Okay. Okay. I want to show you another example of a multipolar type cell, and I'm going to use the cerebellum as an example. Now, what I'm about to show you is what's called a Purkinje cell, this middle picture in this picture from the book. Purkinje, which is a horrible mispronunciation of this person's name, he's just an anatomist from 1800s, I think, 1850, 40, something like that, who was very prolific, which I basically mean had no social life and spent all his time staring through microscopes. There are actually a number of things in anatomy that are named after him because he discovered them, and this being one of them. Now, what we're going to see under the microscope is only the cell body, but understand that coming off the cell body, there are a bunch of dendrites and a pretty significant axon coming off of it down here. Now, the cerebellum is the little brain. When you think of the brain, you probably think of the big wrinkly thing, like the gray matter, white matter picture I showed you before. This big wrinkly thing is the outside part of the brain. This is what we call the cerebrum, which is the Greek word for, Latin word for brain. Okay. There's another piece of the brain which is attached in a sort of inferior posterior position, okay, which in this model that I'm showing you is kind of a tannish color. In actuality, the two pieces of the brain would be the same color, but for the sake of this model, they just colored them different. But this is a cerebellum, which really means little cerebrum. Okay. They both have wrinkles all over their surface. The wrinkles on the cerebrum are much bigger than the wrinkles on the cerebellum. Okay. Now, I'm pointing out the wrinkles because in this micrograph, we're cutting through essentially one of these wrinkles. So this is the edge of the tissue. Okay, this is clear glass, sort of. It's a little bit dirty, but it's glass. And this is tissue. And we can see the edge of the tissue kind of folding in here and folding in a little deeper here, and then the fold comes out. So when we see one of those wrinkles on the surface of the cerebellum, what we're looking at is this crevice right here, basically. So the wrinkles basically make a very convoluted outline or surface of the cerebellum, and that surface looks like what we have here. There are two major regions of the surface of the cerebellum. The outer part that's fairly lightly stained is called the molecular layer. I actually don't quite understand why it's called the molecular layer. I think it was named that before we really understood what molecules were in the modern sense. So it's just an old-fashioned use for the name. But it stains very lightly, okay, which is this area out here. And then the other very significant layer is the darker staining, grainy looking part here. Now I'm using grain on purpose because what we're seeing are little neurons that are staining nice and darkly. They're very small, so they look like little grains of sand. And we call that layer the granular layer, meaning it looks like it's grains of sand. Between those two layers is actually a third layer that's kind of hard to really make out. But we can see these dark spots that line up right on the edge between the molecular layer and the granular layer. Those dark spots are these Purkinje cells. So this is the 4x objective, so we can just see sort of a big swath of tissue. But we really want to go into the 10x version so we can see the edge of the molecular layer here. Okay, so the very bottom corner of this picture is clear glass. But from here to here, this is the molecular layer, which is fairly light in staining. We can see that there are some small, fairly light nuclei staining in there. So there are some cells there, but they're not very big. And then we have this big region of dark dots all over the place, the grains of the granular layer. And then in between, we have some big cells that we can make out. Now, these two happen to be fairly close together, and then there's a big gap until we get to the next one and a medium gap here. They're spaced at kind of irregular or random distances. In 3 dimensions, we would see that it's a fairly tightly packed layer of cells. Not as tightly packed as the granule cells, but they actually are pretty close together. We just happen to be cutting through an area right here where we don't see any other cells, just because we've missed them with the knife. We're going to zoom in and actually look at these 2 cells a little bit closer with the 4DX objective. Now this one's a little bit fuzzy, but we can make out that it has a nucleus in the middle, and then we can see the darker color of the cytoplasm. You may not really make it out, but there's a hint of something sort of poking off this direction. Okay? This one we can see a little bit more clearly. The nucleus is definitely more obvious, and the cytoplasm is a bit more distinct. On the projected version of this, you might not be able to see it so clearly, but if you look at directly on the computer screen, you should see a pretty good representation of the base of a dendrite right here, and a suggestion of a dendrite coming out here maybe, or over here maybe. Okay. On these Purkinje cells, the way that these are stained, we cannot see the axon. The axon's way too small and it's not going to pick up any stain. But the axons come out of this side of the cell and pass deep through the granule layer. Okay. They're going to actually leave the covering of the cerebellum and go to its deep area, which is probably about 2 or 3 centimeters away from the cell body. Okay. So that's, relatively speaking, a long distance for these cells. But these cells, again, are very big because they have relatively long axons. But more importantly, these cells have a huge array of dendrites. And those dendrites extend away from the cell, let me go back to the 10x, and fill up all of the molecular layer, okay? Which we can't see here because we're just staining for the cell bodies. But if we stain in a different way that we can see the entirety of the cell, you can make out dendrite tree. So these are called Purkinje cells. Okay. So just doing a quick image search for them, we can see a number of different representations of them. This is a drawing of a Purkinje cell, which is very similar to the drawing that we have here, except that it's colored in black. What we're looking at here is actually probably a fairly old drawing that was made by this very prolific anatomist, not Purkinje, but another guy from Spain named Santiago Ramon y Cajal. He's like the godfather of cellular anatomy, neuronal anatomy. He wasn't just boring and looked through microscopes all the time. He was actually kind of interesting. He's also fairly well known for developing some important chemical aspects of photographic development. Unfortunately, the chemicals you use to make photographs, especially when you made them back in the 1870s to 1900 or 1901, somewhere in there, that's when he was most active. That chemistry back then, before photography was as mainstream as it is today, those chemicals were toxic to the brain. And so Ramon Cajal is best known for being just freaking batshit crazy when he was older. He basically induced all sorts of neurodegenerative diseases, and when he was old, he was just weird. Now, that's a little unfair representation. He's actually a great anatomist, and there's a lot of things he did that tell us a lot of things about the structure of cells and the nervous system. But this is an example of that kind of drawing we would do. And it's 2 pictures of the same cell, one taken sort of head on and the other one from the side. So it has a very broad array of dendrites, but they're all kind of constrained into a narrow little band when you look at them from the side. Okay, so 2 examples of that. And then the A at the bottom here labels where the axon's coming out. This is sort of an old-fashioned way of doing it. Like I said, this guy did these things like 100 some odd years ago. Nowadays, when we're looking at a cell like this, we're more often going to stain like this. Now, this is a fluorescently stained cell. And what they've done is they've taken a little micro injector and injected a fluorescent dye into the cell. That dye actually associates with the cell membrane. but it looks like it's filling the cell because it's basically coloring the entire outside surface or inside surface of the cell membrane. But it shows us the cell membrane all the way down to the very smallest diameter dendrite branches that we have at the edge. The way that we visualize this, we use a fluorescent microscope, which will excite the dye with a specific wavelength light laser, and it'll make it glow this colored green. But it makes these really amazing kind of pictures. You can see the whole thing. Now, there's a number of other examples here using the same sort of approach. Here we're staining a bunch of cells together. Here's a population of cells. And here we can see they're actually fairly close together. The cell bodies are fairly close together, as opposed to the distance we've seen between cells here. And that's just a matter of how we section this tissue and how we stain it. We don't see them quite as well. But those are the Purkinje cells. Now, I'm not going to spend a lot of time on this, but I do want to point it out. The other type of multipolar cell that the book introduces in that figure with the drawings is called a pyramidal cell. And usually those pyramidal cells are stained in something very similar to that black colored drawing that I showed you from the Spaniard guy. But this is a more modern version of that stain. And it's also much more chemically safe, and people using this don't go crazy so much. But it's called a pyramidal cell because the cell body is shaped like a pyramid or a triangle or a cone, or whatever you want to say. With the pyramidal cell, there's one dendrite that goes straight up from the peak of the pyramid. And then along the base of the pyramid, there's a bunch of other dendrites extending out away from it. We do not have a slide to show you this type of cell in the microscope slides that we have. We just don't have, there is a slide for it, but I don't like it. It's not a very good stain. So I rather go to these good pictures on the internet just to show you what it looks like. But truthfully, I'm not going to show you this under the microscope just because we don't have a good example of that. In this image search, there's some drawings and computer representations and schematic representations of different versions of this type of cell. But these are called pyramidal cells. And they are another type of multipolar cell. Now, that's the neuron stuff that I want to show you. The other thing I want to show you in the micrographs that we have here today are some specific things about glia. Now, I already showed you what's probably an astrocyte in the gray matter of the spinal cord, the satellite cells and Schwann cells associated with the dorsal root ganglion. I want to show you, going back to the spinal cord picture here, ependymal cells make a epithelial-like lining to the empty hole right here in the middle of the spinal cord. Okay. So this is at 4x. If I go to the, oops, that's not the one I want. That's not it. Shoot, where is it? Oh, I labeled it ependymal cell. So the spinal cord ependymal. So this is just a 10x magnification of the picture I was just showing you. And we can see a little bit more clearly that these are individual cells. They kind of look like an epithelium. They are not an epithelium. These are glia, nervous tissue. But in the central nervous system, because of the blood-brain barrier, we have a different type of fluid filling in the extracellular space. And in this hole here, as well as perfusing into the tissue around it a little bit, is this cerebrospinal fluid. That's made by ependymal cells, not these ependymal cells in a different part of the brain. There's ependymal cells that make that fluid specifically, but that's what they look like. And then the other glia I want to show you are represented in 2 or 3 slides. And the 3 slides I'm going to show you here are stained in a very different way from anything we've looked at. These are stained with a osmium-based stain which highlights lipids. Now, osmium is a heavy metal. It has a big atomic nucleus which blocks light, casts shadows. And so everywhere that we see a black spot here, that's where there's a cluster of lipids that are picking up that osmium stain and casting a shadow. What we're actually looking at here is in the periphery, a nerve that has been cut along its length in half. So up at the top, the thing that kind of looks like a watermelon slice, think of that as it used to be a complete nerve, and it's been cut in half so that now it's just the bottom half of that. And then the bottom half has been cut in cross section. So this is one cross section through half that nerve. But beyond the watermelon shaped slice there, everything else here is a different type of tissue. It's not nervous tissue. So what type of tissue are we looking at? Besides the watermelon part at the top corner, all of the rest of the big black spots, what type of tissue is that? So what does the black tell you that we're staining? Lipids. So what type of tissue is going to have big black deposits of lipids?
Adipose.
Right, adipose tissue. So this nerve is passing through a little adipose tissue. It's being cushioned by fat or something like that, perhaps. I'm just doing that to point out what the stain is showing us. The adipose tissue that we've seen in the past is kind of a negative image of this. Okay, this is a lipid stain, essentially, and otherwise we look at things like this with cell stains, that stain cytoplasm and DNA and nuclei and those kinds of things. So everything that we don't see here is what we would see in a cell stain of adipose tissue. So they're sort of negative images. Now this is the 10x objective, and it's zooming in just on that cross section of half the nerve. Now, what we're seeing here again is osmium pointing out where there's lipids. And you can see in some of these, I could have perhaps made a 40x version of this. This is as much as I can zoom in. You can see sort of a ring with a hollow center. And there's a few more over here. And really all of these should be the same thing. What we're looking at is an axon that's been cut in cross-section and wrapped by the lipid-rich myelin insulation. So what we're seeing, the black stuff, is the myelin wrapped around it. And the hollow center in those circles is the axon, the axoplasm. Now to explain this a little bit more, let me go to this picture. Oops, that's not the right one. This picture, well, here, that explains myelination. So this is a Schwann cell, and the way that myelination works is one cell will wrap around an axon, sort of like pigs in a blanket, until there's layers and layers of cell membrane of the Schwann cell wrapped around the axon. That's the insulation. Okay. This is the cytoplasm of the Schwann cell, and as it starts to wrap around, there's cytoplasm in the layers that are wrapping around. But once it fully wraps around, it basically squeezes all the cytoplasm out, so you just have cell membrane, cell membrane, cell membrane, cell membrane, cell membrane, just layered up right up on top of each other. The picture at the bottom is a electron micrograph, and it's stained with that same osmium stain, so that where there's lipids, we see some dark spots, dark staining. Now, we're also staining this with another stain that's based on lead, and lead associates with proteins. So what we're seeing here is a very high resolution image of where there are lipids or proteins in this tissue. And with the kind of resolution we can get from an electron micrograph, we can actually figure out what we're looking at pretty easily. Okay. So the very thick dark line that we see surrounding this, that's the myelin around this axon. Okay. And then inside, the lighter interior, that's the axoplasm inside. All of the little dots that we see in there, those are proteins that are running the length of the axon, and they're picking up some of that lead stain because they're proteins. We can also see a little circle right there and a bigger circle right there. Those are vesicles that are traveling along the length of the axon. So usually in the cytoplasm, certain proteins are being made. They get packaged up into vesicles, and the vesicles physically move down the length of the axon. So the outline that we can see there, the outline that we can see there is just the phospholipid bilayer membrane of the vesicle. We don't see any contents to it. I don't know exactly what's actually in them, but they look like they're kind of empty. The two spots that we see, this one next to the big vesicle and this one off in the corner, those are mitochondria. And mitochondria are very easy to identify in electron micrographs because they have an outer membrane and an inner membrane that's folded up inside of it. So if you look closely at these, or better representations of mitochondria, you can see a line that defines its outside membrane and then another line sort of squiggly all around inside, which is its inner membrane. Mitochondria are very easy to pick out in this tissue. So we see those. Now, I'm not positive, but I think this might be a cell nucleus, but I'm a little surprised. It looks pretty big for a nucleus. I'm not quite sure what it is, but and I can't quite make out the inset there for any more detail. But anyways, this is an electron micrograph showing how osmium stains the layers of cell membrane that are in the myelin. In the projected image on the screen right here, you can't really make it out. But if you look on the computer screen directly, you will definitely see, going right through here, very clear lines that are the individual layers of cell membrane stacked up on top of each other. And you could probably sit there and count them, but there might be about a dozen layers of cell membrane in there. Okay. And that's what myelin is, just Schwann cells or oligodendrocyte membranes wrapping around the outside of an axon. And just makes a very tight seal around the axon in that location. Now, the last slide I want to show you uses the same kind of staining technique, that same osmium stain, but instead of looking at a sectioned nerve, what we're looking at here is a teased nerve. So what was done is, just like in the watermelon slice picture I showed you a second ago, the cell was cut in half and opened up. But instead of making cross sections of that half of a nerve, instead they took tweezers or something and pulled the individual nerve fibers out. So what we're looking at here are individual axons, and they're black because they're wrapped in myelin, and that myelin picks up that osmium stain. Now, what I want to point out in this is little gaps like this here, or that one there, or that one there. There's actually a little gap along the length of the axon between regions of myelin. I didn't point this out when I was showing you the neuron here. Let me pull this back up. So the myelin is represented by this short piece here, and then there's a gap, and then there's another piece of myelin and a gap, another piece of myelin and a gap. Those gaps are very important for how the electrical signal travels along the axon. If I show you an electrical wire like this that's insulated, the insulation is a continuous covering because you don't want the electrical wire exposed where you might touch it and get a shock or something like that. But for an axon, there needs to be a little exposed spot every so often because the electrochemical signal that is the action potential, which we're going to talk about next week, there has to be a place where the axon's membrane interacts with the extracellular fluid. So if it was a continuous insulation running the entire length of the axon, then the action potential wouldn't be able to travel that whole distance. The gaps are spaced apart so that the electrochemical signal can be refreshed along the way. Now, in a picture like this in the textbook, they're always going to make the myelin sheaths, which is what these things are called, look kind of short. But actually, and the reason why I'm showing you this picture is that they're pretty long relative to the thickness of the axon. So here's one right here. That's a gap. They're called a node of Ranvier. This thing right here is not a gap. That's actually a little crushed part of the nerve. And it just doesn't look right. It's where the thing got crushed and the myelin got destroyed and it doesn't stain. So imagine actually that's not there, because from that gap, it should continue on until we get to here. That's the next gap. Okay. So in the drawing from the textbook, they look pretty short. But really, if it was drawn to scale, one myelin sheath would go off the screen. Okay. The gaps are that far apart. If there are so many little myelin sheaths that they're that close together, they wouldn't be very functional. They have to have widespread gaps, which increases the speed that the signal travels along the axon. Okay, so I just want to point out, in real life, that's how far apart they are. Okay. We can see one right at the edge of the screen here for this fiber. If we follow that fiber along, there's its next one. And if we follow it along further, I think it's this. I would have guessed that the next one would have been right about here, so it might be covered up by this fiber, or it might be just off the screen. We can see a gap right there, and if we go up this fiber to this side or down this way, we never get to the next one on that fiber, because whichever direction we go, it would be actually off the screen from the one in the middle there. Those are some good examples of what they look like. I just wanted you to make sure that you see that there is actually quite a bit of different distance between those gaps, which will be important when we talk about how the electric signal travels along the axon, okay, which will be a topic for next week. So this week's folder has a link to the reading, which is this chapter that we've been working on. And then there's part 1, 2, 3, and 4. Okay. And then I just put a link to the slides we were looking at there. Next week's folder, which is week 11, there is no part 1, 2, 3, or 4 assignments. Okay. There is an assignment that says excitable membranes overview. You remember when you did the tissue, was it tissue? No, levels of organization overview when you finish the tissue chapter assignments? Remember that extra assignment with 3 more questions? We have an extra assignment here that's the only chapter assignment in next week's folder. Okay, so this week's chapter assignments, nervous tissue part 1, 2, 3, and 4, are all that we have of chapter assignments for this week and next week. Okay. But then once you finish them, you will have an additional overview assignment here. And that overview assignment is looking at the electrical activity stuff that we're going to talk about next week, which applies to muscle and nervous tissue. So the stuff that we learned in the muscle chapter that has to do with action potentials and excitability and that sort of stuff will be relevant to that assignment. Okay. Now, I just want to point out, I'm showing you this in my instructor view. When you go into this folder, you will see a blurb saying that such an assignment exists, but you won't see that assignment until you finish the nervous tissue part 4 assignment in the previous chapter. They're tied there. It's not tied to the score you get. So if you do it late, you don't have to wait for me to check it off to do the overview assignment. Just completing it will open up the overview assignment. But you won't see it unless you finish the part 4 assignment in there. Okay. And then also in next week's folder is the lab quiz. And like I said, it's partly over the tissue that we talked about tonight, but I'm going to continue talking about neuroanatomy to finish up that stuff. I may not get to the rest of the neuroanatomy next week, and if that's the case, I will tell you, don't bother doing that lab quiz until the following week. But my hope and intention is to get that stuff covered somewhat next week, which kind of depends on how much stuff I get posted online for you to watch outside of class. But so for this chapter, which is this week and next week, you have part 1, 2, 3, and 4 assignments, which are in this week's folder, and an overview assignment, which is in next week's folder. And there's this lab quiz, only lab quiz for this week and next week, which is sort of over the lab material that we're covering this week and next week. So those are the assignments you have. The following week, what's week 12 in this, will be on the anatomy of the nervous system chapter, and it has a part 1, 2, 3, and 4. And that will be the last of the chapter assignments that you have for the semester. The last week, week 13, is on that neurological exam material, and there are no assignments associated with that. It'll just be a week for us to talk about how we can use neurology to understand some of the stuff that we're covering this week, next week, and the week after. So it'll just sort of be a wrap up for the nervous system stuff, but it doesn't have its own set of assignments, which means you have one set of part 1, 2, 3, or 4 assignments for this chapter, and one set of part 1, 2, 3, 4 assignments for the next chapter on the anatomy of the nervous system. That's it for the semester and the overview assignment. That's what you have to look forward to as far as assignments. Wrapping up the semester, things are going to get kind of tough and maybe overwhelming, so I'm sort of assigning fewer things or spreading the work out over more weeks so that it sort of slows down a little bit. As a trade-off for that, the second is practical sucks, so you get that to look forward to. But I'm going to try very hard, and I try every semester and don't quite make it. I'm going to try even harder this semester to prepare you for that as much as possible. And a lot of that should be in that last week of stuff about the neurological exam. So the parts of the practical that suck are the nervous system stuff. And so since they're about case studies, talking about the neurology of the nervous system will hopefully help you understand those cases and be more prepared for it when we get to that. So that's what we have to look forward to. That's it for tonight. Have a nice week. I'll see everybody. Let me say this explicitly. I will see everybody next week. I've had several people say, do we have class next week? Yes, next week is Thanksgiving, but Thanksgiving's on Thursday. It is not a week-long vacation. So we will have class on Tuesday next week. So I'll see everybody next week.